Human penis is composed of 3 cylinders of spongy erectile tissue. During sexual arousal, the erectile tissue fills with blood from the arteries, and that's what causes the hardening or the erection. All right, let's get into gametogenesis. Gametogenesis. How do we make our sperm? How do we make our eggs? How do we make our gametes? The production of gametes differs in male and female, reflecting the distinct structure and function of their gametes. Sperm are small, motile, and must pass from male to female. Eggs are much larger and carry out their function within the female. And if you were to draw sort of a scale to get some idea of, how large is a sperm compared to an egg, it would be something in the order of that, maybe even more of a difference. So you can see sperm are way smaller than the eggs. And this makes sense why sperm are so much smaller than eggs. And what it comes down to is numbers. How many sperm are ejaculated each time, roughly? I told you this in a previous lecture. Around 200 million or so. How many eggs are released each month? Usually 1. So you compare, say, 200 million sperm to 1 ginormous egg, and that's why you get such a size difference there. So how do we make sperm? Spermatogenesis. Think of it this way, sperm genesis. What's Genesis mean? In the Bible, that's the beginning, right? So it's the beginning of sperm. Continuous and prolific. Tell me about it. Prolific. Millions of sperm are produced per day. Each sperm takes about 7 weeks to develop. If you want to become a dad, then make sure your lifestyle is really good, at least, let's say, 3 months prior to trying to have a child, because that's about how long it takes to make some healthy sperm. Oogenesis, the development of a mature egg. So let's look at that. Ooh, those are eggs. Genesis, the beginning of eggs. It's a prolonged process. In males, spermatogenesis, it's pretty simple to understand. But females, they're much more complex. Immature eggs form in the female embryo, but do not complete their development until years or decades later. All right, let's tackle spermatogenesis, the beginning of sperm. Spermatogenesis differs from oogenesis in 3 ways. All 4 products of meiosis develop into sperm. So you basically get 4 haploid sperm. Remember, in meiosis we have 2 cellular divisions, and that's what gives us 4 haploid sperm. In females, only 1 of the 4 becomes an egg. So really, you just have that 1 oocyte, haploid oocyte, and then the polar bodies, which we'll speak of in a sec. Spermatogenesis occurs throughout adolescence and adulthood. So think of it this way. My 9-year-old boy, he's 9 at the time of recording here, hopefully he's not producing any sperm yet. I don't think so. Puberty has not quite kicked in yet. But once puberty kicks in, then he will begin producing sperm. And how long will he produce sperm for? That's right, the rest of his life. Crazy. So you can have a, sorry, I don't know if that was funny or not, but this thing seems to think so. Isn't that crazy, though, that you could have a 104-year-old dad I know it's weird. You may have thrown up a little in your mouth there, but even at 100 years old, it is possible that a man could get someone pregnant. Now, obviously, sperm probably not going to be as healthy. Sperm count probably going to be down quite a bit. And, of course, other issues that could happen there. But, technically, it is possible. Now, moving on. Where did my pen go? 1 second here. Spermatogenesis occurs throughout adolescence and adulthood, as we said. Sperm are produced continuously without the prolonged interruptions in oogenesis. So let's tackle this process here. Now, for my class, we are not going to cover the spermatogonium or the spermatogonial stem cell. We are going to pick it up here at the 2N primary spermatocyte. Okay, this is where I like to begin with general biology. So we've got the primary spermatocyte. Notice that it is 2N, which means it is diploid. Both spermatogenesis and oogenesis always begin as a diploid cell, a somatic cell, a body cell. Okay? Now, this primary spermatocyte will undergo meiosis 1, and that will result in 2 haploid secondary spermatocytes. What I like to do in my classroom is to keep track of the chromosomes. If this is a human, how many chromosomes are in a diploid cell? That's right, 46. And once you go haploid, how many are there? 23. 23. Notice how after meiosis 1, we are now haploid for the rest of it. Next up, after the secondary spermatocyte undergoes meiosis 2, now we have 4 haploid, and we call those spermatids. Okay? Early spermatids, and each one of those are haploid, so 23, 23, 23, and 23. So all 4 of those will then mature and metamorphosize into sperm cells with help from the Sertoli cells. So now you've got your sperm cells and they're ready to swim. Those are 4 haploid sperm cells. If we look at what a sperm looks like, there's not much to it. It has a tail, it has the midpiece, and there's a bunch of mitochondria in there. Mitochondria are providing that energy so that it can swim its epic distance. And inside the head, we have the nucleus. So inside that nucleus are 23 single chromosomes from your dad. And we have the acrosome. That's right in the head of the sperm. And that has some digestive juices which are going to come in handy if that sperm is lucky enough to reach that oocyte. It can help to digest the protective outer covering, the zona pellucida. All right, so that's spermatogenesis, and this is happening from puberty all the way until death. Now, something that I spoke to you earlier about is that in meiosis, when you're making sperm and when you're making eggs, every single sperm or egg that has ever been made is unique. Now, that is pretty mind-blowing. So I like to bring up this idea, and it may make you throw up a little bit in your mouth, but let's give it a go. The idea is that imagine all of the ejaculations that have ever happened in the history of humankind in the last 500,000 years. Okay, so that's kind of gross. But none of the sperm have ever been the same. the chance is about 1 in 70 trillion that you have two sperm that are the same, and the same goes for all of the eggs that have ever been made by females in the past 500,000 years. None of those have ever been the same. They're all unique. Isn't that crazy? I mean, I should put a little asterisk beside that, because with cloning, it is possible to have two sperm that are identical or two eggs that are identical. All right, so wipe yourself off from that little vomit, and let's move on to oogenesis. Oogenesis, a little more complicated, those females. All right, let's take a look at why is it more complex. Well, first of all, there's something really fascinating here. Did you know that all of the eggs are actually manufactured while you are a fetus? So as you're developing inside your mama's belly, that's when a female is building all of her oocytes for later. Because of that, it's even more important, I would say, for someone who wants to be a mom to take control of their lifestyle and try and live a really healthy lifestyle, let's say at least a year before you want to become pregnant, and especially during your pregnancy, of course, very important to take good care of yourself. And the way to think of it is this: you could have effects on your grandchildren based on the lifestyle choices you make while you're pregnant. If you're having a daughter, then your daughter's eggs are being made while she's inside you. So just think about that and make good, healthy decisions, including things like managing your stress levels, and that can ensure healthy development of your daughter's oocytes. All right, now, once a female is born, those oocytes are now kind of frozen in time at prophase 1 of meiosis. So they only just kind of started meiosis, then they stop. They're arrested until puberty kicks in. And then when puberty kicks in, then each month one of the follicles will be chosen, the chosen one, to release and ovulate. And that's when meiosis will then start back up again. And then here's another weird thing. Meiosis during oogenesis will never be completed unless a sperm fertilizes that egg. So the only way that Meiosis Two will get finished in oogenesis is if a sperm fertilizes that egg. Another thing to keep in mind with oogenesis is it only takes place until menopause, around age 50, plus or minus 10 years roughly. There's a lot of variance of when it takes place. But once menopause has fully hit, then the body gets hormonal changes, which results in not releasing any more eggs each month. All right, so now that we got all that out of the way, let's give you some more complex stuff about oogenesis. Again, in my classroom, we are going to pick it up here at the primary oocyte, and just like in spermatogenesis, we start with a diploid somatic cell. How many chromosomes does that have? That's right, 46. Almost did 23 there. That would have been bad. So 46 chromosomes over here. And we are starting here at a primary oocyte, present at birth, arrested in prophase of metaphase 1. Now, look at this weird thing that happens. Instead of having an equal division like we did in spermatogenesis, we have an unequal division. So in females like to put all their eggs in one basket. Yes. What they do is they want to preserve as much cytoplasm as possible in this cell, and that way when they're only making one egg total to be exported, then it's going to have all the nutrients needed. All of this is cytoplasm. That has organelles. It has all the nutrients needed for that developing zygote. And one of the reasons is that the developing zygote, once fertilization takes place, it will not implant into the mother's uterine lining for around 5 days or so. So you can think of it this way. It's kind of like a smoothie to help the development of the zygote as it's floating along through the oviduct, making its way through the cilia, and eventually implanting into the uterine lining. And that is where it can tap into mama's blood supply with the development of the placenta. So now you can understand why there's unequal divisions here. You want to preserve as much cytoplasm and nutrients and organelles as you can. And then the other one, what happens to that? It's called a first polar body. That polar body does have chromosomes. It has 23 chromosomes. And the secondary oocyte, which had just completed meiosis 1, it also has 23. So what do you do with that first polar body? There is not a great consensus on what happens there. Some people say it's just gotten rid of. Other people say that it gets reabsorbed. So I'm not really sure yet. If you know, please leave a comment below. But really, the first polar body is just 23 chromosomes and not much else. Then we will move on to Meiosis 2. But remember what I said. Meiosis 2 can only happen if sperm enters. If the sperm enters, Meiosis 2 will be completed, and that will result in another polar body. This first polar body can also split and make another polar body. And then you've got your fertilized egg. At that point, okay, let's still do our math. 23 there, 23 here, 23 in the sperm, 23 in the sperm. Notice that the sperm and the nucleus of the egg here have not joined yet. Once the nucleus of the sperm and the nucleus of the egg join together, boom, now we've got a zygote. Now we've restored diploid status, and now we're back to 46 chromosomes for the developing zygote. At that point, once you have a single-celled zygote, that zygote will now switch over to mitosis, and mitosis is going to result in a doubling of cells until eventually you get to adult size, which is going to be around, say, 30 trillion cells. Science. Whoops, wrong side. Science. Oops, the wrong side. Science. Right here. Boom. All right. So, oogenesis, pretty complex. Let's look at some other complexities here of oogenesis. You've got the primary oocyte, and then look, it's within the follicule. So the follicule is this big area. It's a support cell to the primary oocyte, and with certain hormones, you'll see changes. So you've got the maturing follicle. It's about to be ovulated, and then it pops out here, goes, makes that noise, and pops out. There's actually a funny term here. It's called Mittelschmerz, and that is the word for when you release that egg and ovulate it. Some females claim that they can actually feel this happen. So middle smirts release, ovulate, but then notice that the corpus luteum, that was where the egg popped out of, that is now going to strengthen up, and it's going to have hormonal effects which will then help to support the developing pregnancy, if there is a pregnancy. Now, what is going on over here with the degenerating corpus luteum? That would be if pregnancy did not take place, then the corpus luteum will degenerate quickly. So I believe it's the corpus luteum is very helpful for the first trimester of the pregnancy, and at the second and third trimesters, at that point, I don't think it's as important.